Hi everyone, my name is Sharon Vijayanand. I'm a third year graduate student at Mercer University College of Pharmacy in Atlanta. Today I'm going to be presenting my research, which is titled No Pain, More Gain, Microneedle Vaccines for COVID-19 and Influenza. SARS-CoV-2 and influenza are contagious respiratory viruses, and as of now, for influenza, yearly immunization is required. SARS-CoV-2 may also be the same way with frequent immunizations. The goal of this study was to develop and test microneedle-based vaccines for both influenza and COVID-19. Our vaccine approach involves the use of polymeric microparticles as the carrier for the vaccine antigen. Our immunization strategy involves using dissolving microneedles, and the advantage of using microneedles is that they dissolve to release the vaccine microparticles into the epidermal layer of the skin, which is then actively taken up by the circulating dendritic cells. Influenza as well as SARS-CoV-2 have several antigens of interest, but for this study we chose the matrix 2 ectodermal protein on the influenza virus because the M2E protein is highly conserved across the influenza A strains, which makes it a perfect candidate to test any cross-protective responses. And for SARS, we chose the heat-inactivated SARS-CoV-2 virus as we were starting it off as a pilot study. The objectives of these studies was to formulate and characterize the corresponding vaccine microparticles, and then we wanted to prepare and characterize the microneedles and do an in vivo assessment study of our microneedles. Moving on to our methods, our vaccine microparticles were formulated using a double emulsion method and lyophilized to form a dry product, which was then characterized for the following parameters as shown here. The microneedles were prepared using a spin casting method using a very basic hyaluronic acid and sugar gel base. We then characterized the microneedles, and we also tested the in vitro pore formation using murine and porcine skin. We also wanted to see the in vivo pore formation in live murine skin as well. We then prepared the vaccine-loaded microneedles and immunized our mice, following which we analyzed the serum antibody levels using ELISA, followed by challenging the mice with a live influenza virus and organ isolation and analysis. As part of the characterization, we observed the microneedles under the digital microscope as well as the scanning electron microscope, and we found that the needle length of the 8x8 array was about 520 micrometer, and for the 10x10 array it was about 370 micrometer. Following application on murine skin, the microneedles quickly dissolved within 5 minutes, and that's the SEM image before and after application that's shown here. We then cross-sectioned the murine and porcine skin to see how the pore formation looked under the microscope. As you can see here, the red arrows indicate the pore formation following application of the microneedles in skin. We tested the in vivo pore formation in live murine skin by administering indocyanine green loaded PLGA microparticles, which were administered using microneedles. As you can see, within 10 minutes, there was an increased green fluorescence intensity, which then gradually reduced with time. We plotted the green intensity value versus a time graph, and we found that the intensity decreases with time, and increased intensity did correspond to like successful pore formation in live murine skin, and the decrease in intensity may also indicate the pore closure after a certain time. The timeline of our influenza study is as follows. At week 0, there was a prime dose. At week 3, a booster dose. And at week 6, we did a live viral challenge, following which we sacrificed our animals at week 9. We then assessed the serum antibody levels at week 1, 4, and 7. And as you can see from the graphs displayed here, there were significant responses that we obtained. So the observations that we wanted to note were we got responses that were comparable to the flu zone, which is the marketed flu vaccine. And we obtained significant responses as compared to the naive group. M2E specific antibodies were produced from these graphs. And antibody levels remained significant following viral challenge with a Philippines H3N2 strain. As you can see, the challenge was done in week 6, and the week 7, the serum antibody levels still remained high. We also observed that there was a balanced IgG1 and 2A response. The response of IgG1 and 2A were not skewed in any direction. Significant IgA response was also observed. The timeline of our SARS-CoV-2 study was a little bit different from the influenza study. So at week 0, we had a prime dose. At week 3, a first booster dose, and we had another booster dose at week 5. There was no viral challenge that was done. And at week 10, we sacrificed the animals for further analysis. We then assessed the antibody levels, anti-SARS-CoV-2 specific antibody levels using ELISA, and we also observed significant responses as we did with the previous study. So the key observations to note here is that we did observe significant response compared to the naive group. SARS-CoV-2 specific antibodies were obtained. IgG level was significant. IgG1 level was significant for all the weeks. However, IgG2A level was significant only at week 8. Significant IgA responses were also obtained for this study. As I mentioned earlier, no viral challenge was done, and that was a major difference between the previous study and this study. In conclusion, the microneedle vaccines were prepared and characterized. The microneedle vaccination induced virus-specific IgG and IgA antibodies for both the studies. IgG1 levels were tested as they indicate a Th2-mediated helper T-cell response, whereas an IgG2A level response indicates a Th1-mediated cytotoxic T-cell response. And dissolving microneedles is an attractive option for vaccination because it's pain-free, it's appealing to the pediatric and the geriatric patients. It can be self-administered in case of adults, and it may potentially overcome problems due to cold chain storage and vaccine distribution. As for our future studies, we plan to run some neutralization assays, also look at cell-mediated immune responses, and we also want to test a combination vaccine study where COVID-19 and influenza vaccines are given as one microneedle vaccine patch. With that, I would like to conclude my presentation and thank my major advisor, my co-author collaborators, and all other contributors for this study. I would like to thank you all for patiently listening to me today and also for this wonderful opportunity to present my research, and I'm open for questions.